So welcome to our revision tutorial looking at enzymes and how they work within the body, especially in relation to digestion. So the first thing to remember about an enzyme is that it is a biological catalyst. Now, in simple terms, all that means is that enzymes help to speed up biological reactions. Reactions within organisms, within the body. So, with that in mind, we're going to have a look specifically at the action of enzymes within the digestive system. But before we do that, it's worth just looking at the general process of how an enzyme acts on a substance. So we've got a little diagram here that talks about how an enzyme helps within the process of digestion to break down a substance into smaller pieces. So here we've got our enzyme. Now, our enzyme is a unique molecule, okay? And within the molecule, it has a unique shape called the active site. So as we said, it's important to bear in mind that these are unique. Now, what happens at the active site is where substances are broken down. Now, these substances are often referred to as substrate molecules. Now, these substrate molecules are specific to the active site. So they are a complementary shape, which means they fit really easily into the active site. So a bit like a lock and a key, okay? The substrate acts a bit like a key, which is only unique and will only work within a specific lock. And the active site acts a bit like the lock. It only accepts specific shaped substrate molecule moving into it. So the active site and the substrate bind to each other to form an enzyme substrate complex. So they bind and they form what's called an enzyme substrate complex. This is just the molecule, substrate molecule, and the enzyme bonded together. Then the enzyme then acts to break down the substrate into separate products that are often smaller. So we've got products. Now, this breaking down to form products is often referred to as a catabolic reaction, which is the breaking down, the opposite of which is an anabolic reaction. For the sake of this is a catabolic reaction. And the important thing to bear in mind is that the enzyme, in terms of this reaction, is not used up. So it can be used again to act on other substrate molecules to break them down into new products. So in general, that's how an enzyme works. But there comes a point there come points where actually this can be affected so that it doesn't work as well. Now, this process, where it doesn't work as well, of this bonding of the active site and the substrate molecule to form new products, is called denaturing. So denaturing specifically is when the active site changes shape so it can't bind to specific substrate molecules. So this is called denaturing, and this is often when enzymes are put into conditions where the pH or temperature surrounding them in their environment has changed. So pH and temperature can often have a major impact on the shape of an enzyme's active site. So normally, substrate molecule can bind to the enzyme and it works absolutely fine. However, if it undergoes denaturing, so this is a denatured active site, the enzyme can't bind to the substrate molecule. So that means products can't be formed. And that's how we end up with denatured enzymes, and denatured enzymes have an impact on the ability of a normal enzyme to bond with a substrate molecule at its active site and break it down into new products. Now, in terms of digestion, there are three main enzymes to be familiar with and consider. So the first enzyme is amylase, okay? Now, similar to the processes we just talked about, amylase is specific for breaking down starch. So our substrate is starch. So the amylase bonds to the starch molecules and breaks them down to form our product, which is glucose. So if you think about it in terms of food groups, when you eat particularly starchy foods such as pasta, bread, potatoes, what happens is amylase produced in your salivary glands acts on the starch molecules that make up those foods, those starchy foods, and break the starch down into glucose that can be used within the body. Similarly, if we think about proteins, dairy, meats, things like that, they are acted on by enzymes that are referred to as proteases. So, enzyme, protease. So the protease acts on substrate molecules that are proteins and break them down into smaller parts, constituent parts that they are made up of, which are products, which are amino acids. So what that basically means is when you are eating proteins such as meats, if you think about people who bodybuild or get more muscle, they're often told to eat a lot of protein, and that's because the protein is taken in by their body, digested down, and broken up into amino acids that can then be reformed into new protein, which can be used to build new muscle and things like that within the body. So that's protein being broken down by protease into amino acids. And then our final example is fats. Now, fats are often referred to as lipids. So our substrate is fats or lipids, which obviously contain quite a few fatty foods, butter, dairy, chocolate, any such sorts. Now they bind with our enzyme lipase. So our enzyme here is lipase, and the lipase acts to break down the fat into its two constituent parts, which type of sugar called glycerol and fatty acids. So the products that are produced when we digest fat from the food that we eat are glycerol and fatty acids. So they're just three examples of the actions of enzymes within the digestive system that you'd be expected to know. The name of the enzyme, the substrate that it bonds to, and the product that it produces. So thank you for watching our little tutorial on enzymes.